Morning. Happy Valentine's Day. It's not that I remember all these holidays, but you have a school-aged kid, they will remind you of all these things. Anyway, so welcome to SBMI. As Susan mentioned, my area is in bioinformatics. And so I will talk a little bit about what is bioinformatics, and I will show you one project that we have been doing, which actually use the literature and the information science try to make new discoveries. So what is bioinformatics? So by the way, does anybody know NCBI? Right? Yeah. So this is within the National Library of Medicine, which actually supports a lot of libraries around the nation. National Center of Biotechnology Information is the place where actually a lot of early bioinformatics work are done. And so they have this official definition, which I think most people agree. So basically, bioinformatics is the field of science in which biology, computer science, and information technology merge to form a single discipline. The ultimate goal of the field is to enable the discovery of new biological insights, as well as to create a global perspective from which underlying unifying principles in biology can be discerned. So basically from this, you can see that, first, bioinformatics is a cross-disciplinary, which involves biology, computer science, and information technology. And also the goal is really to make new discoveries, and that can help us to have a deep insight into biology, which can in turn help us to develop, better medicines and therapeutic strategies and things like that. So practically, what is bioinformatics? If you just look at the word, right, it's bio plus informatics. Basically, it's an application of information technology in biological research. In general, bioinformatics covers 3 different areas: information management, data analysis and mining, and data dissemination. I will talk, a little about all these different areas. So the first area is information management. Basically, as you may know, 10 years ago, we have finished the human genome sequence, and during the process of, generating the human genome sequence, there, a lot of high-throughput methodology are developed, and those high-throughput technology analyze the biological samples and generate a huge amount of data. And through that process, the needs to cope with that huge amount of data, increases dramatically, and that includes, how to gather the raw data from the lab, from the experiments, and format the data, store the data, manage the data, and integrate the data, relate them together, and also how to communicate the data among different investigators, and also how to develop the standards to annotate the data, things like ontology. And couple of examples that, in terms of bioinformatics research, something, for example, like, develop a new schema for the database to deal with large amount of data, or, to develop efficient methods to compress the data to transmit, the large amount of data that generated from biological experiments through the internet. The other important part of the bioinformatics is the data analysis and data mining. And as huge amount of data are generated, how to identify useful information from this huge amount of data is challenging. As you have more data, you have more noise as well, right? So, that's sort of demand us to develop new methodology for data analysis and data mining. So the theoretical foundation for this kind of data analysis and data mining is from the mathematics, statistics, and the computer science. And also, we cannot forget about the domain knowledge, which is, biology and biomedicine. Because without the domain knowledge, you can apply fancy algorithms, but you might really look into the wrong questions. So the other thing about the data analysis and data mining is that we are dealing with large-scale data analysis. For example, the methodology that is used here, they will have hundreds of thousands of data points or even millions of data points. And so, typical simple statistical analysis like, t-test is not going to be sufficient for analyzing this kind of data. You have to develop, new methods. And another challenge here is that because, running these experiments to generate the data is expensive, and a lot of times, people can only make, very few repetitions. So, for example, they're measuring 100,000 data points, but they can only repeat like 2 or 3 times. So, that is a significant challenge for the existing statistical approach, which relies on, the large sample size and a few features. The other thing is, to analyze this data, we're also dealing with the, integration, the big data. Nowadays, people analyze not a single feature of the, biological system or single aspect of a patient treatment. But, we're looking at all different aspects, So this is what's so-called big data, We're looking, as Susan just showed, for the biomedical informatics, you can see that we are looking at all the way from molecular level data up to the population level data. Okay, so how to integrate this data together and to sort of mine this data and gain the new insight and find, say, the cure for disease needs a new methodology development. And also, certainly, with all this available information, we're looking into the inference and the prediction. Meaning, if you look at the data for a particular patient, we want to see, whether the patient is prone to a particular disease, is predisposed for a particular disease, or not, based on their, personal genome information. And also, we want to be predictive. For example, if this patient has this type of molecular profile, whether he will be better, be more responsive to a particular drug. And also for a particular cancer patient, if there's certain type of genes or proteins are expressed, whether that's an indication that the patient can survive longer or not. So things researches along this line are things like, for example, we have been doing the data mining, try to identify what are the molecular signatures for cancer, and also whether we can use this cancer to develop the biomarkers to predict whether the cancer patient can survive or not. With regard to the data analysis part of the bioinformatics, there is a significant paradigm shift from data generation to data analysis. As I mentioned earlier, nowadays people use a lot of high throughput technology. Generating data, analyze some patient sample to generate the data is really fast. We rely on a lot of robots and the machines to perform this kind of analysis, and a lot of data can be generated rather quickly and cheaply. But how to analyze the data to gain the insight from this data is a significant challenge. So that's different from in the old days. It takes months or even years to knock out the mice to generate, a little data, and then, the data analysis is fairly easy. So, to deal with this large amount of data and to sort of adapt to this paradigm shift, the idea is very critical, in terms of how to analyze the data, how we can develop new methods to gain new insight. And another important thing with this paradigm shift is that, you can, nowadays, you don't have to do experiments, or you don't have to be in a wet lab. You can be just sitting in your office. You have instant access to a huge amount of data. And you can, if you have good ideas, you can perform analysis, you can make new discoveries. So that is actually a very, I think, maybe something if the librarian are interested, they can do. You can analyze the data, without doing experiments or things like that. But certainly with this kind of analysis, when you make new discovery from the data analysis, experimental validation is important. So the third part of the bioinformatics is the data presentation and dissemination. So basically, as I mentioned, when we have large amount of data, we analyze the data, we make new discoveries, how to make this new discovery available to other people is also a very hot area of bioinformatics research for several reasons. One is that we have huge amount of data. When we make new discoveries, we can probably also will make new discoveries in different areas. And then the second thing is the complexity of the data makes it a challenge to present your discovery as well as the data to other people. But certainly new technology helps. Internet, the web interface, this will make the data very easily accessible, make the new discovery results easily accessible to other people. And also the better hardware then make the rendering huge amount of data on your desktop much faster and easier. And so one particular thing in terms of the data dissemination is the visualization, which as I mentioned, you have large amount of data that has complex relationship. How to visualize all this data is a challenge, and there are a lot of research that's been going on in that area. And so example of this is we try to sort of build a 3D genome model to visualize the whole genome in a 3D space on your desktop. And so that's something that we have been doing, which uses these multi-scale models to visualize complex data of the human genome at a different level. So, so when we talk about the bioinformatics, we also hear probably a lot about another term. It's called computational biology. Basically, the field of bioinformatics evolved, such that most of the present tasks now involve the analysis and the interpretation of various types of data, including nucleotides, amino acid sequence, protein domains, and protein structures. The actual process of analysis, interpreting data part is referred to as computational biology. But typically, a lot of people use these two terms interchangeably. The driving force, I think I covered this a little bit of bioinformatics, is really two sides. One is the rapid accumulation of biological data through these high throughput technologies, and also the increase of the breadth of biological research. Nowadays, people use the high throughput technology not only investigate the molecular level feature of cells, but also to analyze the patient samples and to incorporate the molecular level information into the patient treatment. So all these accumulates a lot of data from the molecular level all the way to the patient and population level. Then the other side of the driving force is the advancement in the information technology. That includes the cheaper and faster hardware, better software, and the new technologies. And obviously, these two are both needed really to advance bioinformatics. Without the big data, there's really no need for the developing new methodology for data analysis and things like that. Without this new advancement in information technology, I don't think we can really deal with the problem that caused by this huge amount of data. So, with this, the paradigm shift for biomedical research from data generation to data analysis, now also there's a shift in biological research. In the old days, most of the biomedical research are what's called hypothesis-driven. So basically, you sort of, so for a well-trained biomedical researcher, they will look into a significant problem, and then they will generate some hypothesis, either a disease mechanism or a solution to treat a disease or something like that. And then, based on that hypothesis, they will design the experiments, and then they will perform the experiments in the wet lab to test their hypothesis. And once they perform their experiments, they generate the data, and then they analyze the data. And based on analysis results, they will see, check with their hypothesis, see whether the hypothesis is correct or not. And then they might modify their hypothesis and continue the cycle until, they have, the hypothesis that's been correct and tested and validated by the experiments. Nowadays, though, with the huge amount of data that's available, so there are a lot of people doing what's called a biology dry lab. That is, it's an informatics approach that try to make new discoveries from the available data instead of doing experiments. And there are two types of approaches of doing this. The first one is still like the traditional hypothesis-driven methods, but it's go to in a different direction. So we look into a significant problem again, and then we probably generate some hypothesis, and then instead of doing experiments, we actually go to the publicly available data and collect relevant data, analyze the data, and see whether our hypothesis is correct or not. And then if, say, we did make a new discovery, and then we collaborate with the bench scientists to design experiments to validate the new discovery. Okay, and then there's another slightly different approach, which is called a discovery-driven approach. Basically, in this case, we are still interested in significant problems, but we might not have a really good idea about, about, say, what's the disease mechanism, or what's the solution to treat a patient. Basically, we started with, say, okay, let's gather all the relevant data and analyze the data, see what we can find. Okay, and then we analyze the relevant data, and then from the data analysis, we might make some new discoveries and generate some hypotheses. And in the meantime, we might take the new discoveries and work with the bench scientists to develop some experiments for validation and for testing. And that will validate the hypothesis we generated. Most bioinformatics groups are really doing this type of research, even though we still have a significant efforts in collaborating with bench scientists to perform the data analysis. So next, I'll talk a little bit about the little project we're doing that basically tries to use the literature to make new discovery. And so, as we all know, librarians, right? So the growth of peer-reviewed publications are really, is at a very rapid pace, and this is only to 2009, but it grows even faster in recent years. So in the old days, when scientists working in a particular area, they probably read all the relevant literatures in that area. They're experts, they know in and outs in that area. And actually, I'm fortunate to have the opportunity to collaborate with someone like that. You sit in a group meeting and somebody come up with something, and the person will just say, oh, in 1977, somebody published a paper about that particular thing. Okay, so it's pretty amazing. But with the the growth of the peer-reviewed publication like that, I think probably it's getting more and more difficult to be someone like that, right? So developing computational methods to help people to analyze the literature and to make a new discovery would be something that can be quite useful under this situation. And also, another reason I'm doing this project is that you can see as a graduate student, I have to read that many papers for one semester. And I actually really promised myself that someday I'll see whether I can do something about it so I don't have to do it anymore. And so that's why I get into these develop computational methods to analyze the literature, and so I don't have to really read all those papers. So how to make a new discovery from literature? So one simple thing, is people can sort of take the literature, an article, a web page, or something, and then you just basically counting the words in that publication, and look at the frequency and see what those words can tell you. So this is one example. During the 2012 campaign, you take the top 10 web pages from Google search about Obama and Romney. You just then take the words from the or phrase from this web page and just simply count them. And as you can see, this word counting indeed can provide some kind of information, right? Things like Chicago, Senate, Illinois, and law, those kind of things about Obama, and things like Republican, Boston, and Bain, those about Romney. So it's a very primitive way of analyzing the literature and to gain some information. But but it's not a really a sophisticated way that can get a lot of information. Not every word can make contribution. As you can see, a lot of general words there. So those things that's not really tell you much. And the other thing is the word frequency is a very rough measure. So there are high frequency words there, but some of the irrelevant words also has very high frequency. So so we have to sort of develop a better way to mine the literature. So one thing we have been looking into, and many people are also doing this along this line, is try to use the ontology to mine biomedical literature. And for several reasons. One is that the biomedical literature is really growing fast, huge amount, and with the diverse, covers very diverse areas, and to analyze those literature is kind of difficult. And so things particularly like, inconsistent terminology. One person will call this disease one way, the other person call the disease the same disease another way. if you try to develop a computer program to analyze these, and okay, thank you. The computer does really not know, that's the same disease called by different names by different people in different publications. And the ontology, can sort of solve that problem because it's well-developed, consistent terminology with structure. I'll get more to that later. And also, certainly, use ontology to mine the biomedical literature can take the advantage of knowledge that's captured in ontology. So what is ontology? An ontology is a specification of a conceptualization. So basically, it's a description of concepts and the relationships. For example, people develop a gene ontology, which basically describe the gene functions and what the biological process the genes involved in, what the molecular functions the genes has, and where in the cell the gene will function. And also, when people have the concepts for all these, they also have relationships among these concepts. So this gives people some ideas about what the gene would do and in what location and how these might be related. And so using this kind of information, you basically can sort of label a gene, tells people what it does, and things like that. So what we try to develop is what's called the ontology fingerprints. So basically, we took the gene ontology, which captures the gene functions and things like that, and then we ask the question: in all the publications about the particular gene, how often each of those concepts in the gene ontology are mentioned? Okay, and so by doing that, we sort of generate these ontology fingerprints. Basically, it's a set of gene ontology terms overrepresented in the PubMed abstracts linked to a gene or disease, along with these terms corresponding enrichment p-value. It is a comprehensive characterization of genes and disease. So I will show you some examples of that. So this is a basic idea behind the ontology fingerprints. All these analyses are based on what's called a hypergeometric enrichment test. So basically, you get all the PubMed abstracts relevant to a particular gene, and all the PubMed abstracts relevant to a particular ontology term. And then you also get the other abstracts that have nothing to do with that gene, and other abstracts have nothing to do with this specific term. So with these, then you can perform this hypergeometric enrichment test. Basically tells you to what degree a term is enriched or overrepresented in the abstract about that particular gene. And what you end up with is what's called the p-value. The smaller the p-value, the more significant this term is overrepresented in the literature, in the PubMed abstracts about that particular gene. So this is the ontology fingerprints for these particular genes called upper lipoprotein C2. And so you can see here are the ontology terms, and here are the p-values. And from the ontology term, you can get some ideas about what the gene does. Okay, and also the p-value will give you some quantitative measure about what of these terms might be more relevant to this gene than the others. So we have over 200 of these terms for this particular gene. So we can also develop these fingerprints for the phenotypes. For example, this is a high density lipids, HDL. And so we can generate ontology fingerprints for these HDL. and together with the p-value. So with this analysis, we basically have a comprehensive characterizations about genes and about the disease phenotypes from the literature. So we want to use these to make new discoveries. So the hypothesis we have now is that we think there are identified components that exist for a given biological pathway. So for those who don't know what a pathway is, basically, you know, imagine humans have 25,000 genes, and these genes are not work alone. You know, they work together, just like, you know, we work in a library, work together. Different persons have different functions, right? They sort of interact with each other to fulfill the function of a library. In the cell, you know, you have many genes. They also work together, but they also work together as a group, you know, like a department. You know, you have different departments, have different people, different departments have different functions. So a pathway is like a department that will fulfill a particular function. And now through the biological investigation, the experiments, and high-throughput methods and the informatics methods, people identify, you know, the components of these pathways or departments. But, you know, we always think, you know, this kind of analysis is not complete, and we want to find the additional components for that, you know, pathway or department, so we can have a better understanding of how genes function. And then that might lead to a better way of treating disease. So, to sort of test that hypothesis, we started with a pathway called sphingolipid pathway. So it plays important roles in tumor genesis and cancer progression. And right now, people have a good understanding of sphingolipid pathways and identify the many genes for that pathway. But we think there are additional genes that might influence the activity of this pathway. So our goal is to use the methods we developed to discover these new genes. And what we try to do is that we take ontology fingerprints between genes, we compare them, and develop a similarity score. This gives us a network that tells how these genes are related. And then we sort of analyze this network, try to identify the new components of this pathway. And indeed, using this approach, we identified 14 new genes that there's no previous publication says these genes have something to do with this pathway. But when we perform the experiments, we indeed, you know, these genes are really important players of this pathway. So, by, you know, showing this example, basically we're saying that using ontology to mine biomedical literature can provide novel insight into biological pathway. And the literature mining and data analysis could initiate new biological investigation without expensive wet lab experiments. And the paradigm shift from the data generation to data analysis in biomedical research could open up new avenue and opportunities for library and literature science. With that, I just want to acknowledge my students and my collaborators. And currently, I received funding from National Institute of Health and the National Science Foundation for projects mentioned. Thank you.